All right. Hi, everyone. Thank you so much for joining our IHC workshop today, which will be hosted by Elizabeth Berry. Before we begin the workshop, I just quickly want to introduce myself. My name is Afrida Rahman-Enyart, and I'm a scientific liaison here at Proteintech. I just want to share a few company slides so that you know who we are as a company. So, Proteintech is a leading manufacturer of antibodies and immunoassays. We have antibodies against over 13,000 targets. We also offer antibody labeling kits, ELISA kits, IHC kits, and cell separation kits. Within our Proteintech brand, we also have our Humankind line, and Humankind manufactures cytokines and growth factors. And all our proteins are expressed in human HEC293 cells, meaning that you can be confident that they have native folding, post-translational modifications, and glycosylation. The third line that we have within our Proteintech brand is Chromotech, and Chromotech manufactures nanobody-based reagents that are ideal for immunoprecipitations, immunofluorescence, and protein purification. What makes us different from other companies is that we manufacture all our products in-house and sell directly to the customer, meaning that you won't see our Proteintech products under the name of any other company. We're also very transparent about our product performance, and we include original validation data. I also quickly want to point out the success of our customers because our customers' success is our success. And Proteintech is really proud that we've actually been cited. It says 160,000, but now it's over 170,000 publications worldwide. And we also won the Researcher's Choice Award from CiteAb this year, showing that our customers really do value our products. So with that, we'll go ahead and begin the workshop. So today the workshop will be led by Elizabeth Berry. And Elizabeth is a PhD candidate at New York Medical College. You might know her from her Instagram handle at nature.neuroscience.phd. And as a graduate student, Elizabeth is studying the impact of opioid addiction on neurons of the lateral hypothalamus. And she's gained a lot of technical expertise while in graduate school, including whole cell patch clamp electrophysiology, IHC, and IF. And Elizabeth hosted this workshop earlier today, and it was absolutely phenomenal. So I'm really excited for her to host it again for us. So with that, I'll go ahead and let Elizabeth begin. During her talk, if you have any questions, feel free to put them into the chat or the Q&A session, and we will address those at the very end.
Awesome. Thanks so much. So let's get started for another round of a interesting topic that we're going to be addressing today. So I'm a final year PhD candidate at New York Med, and my PhD has been focused on neuroscience. So I recently have a lot of expertise in brain histology, IF, and IHC. But previously in undergrad, that's where I started working on IHC and IF. I actually worked on a completely different model, which is a plant model called Piscomytrilla patens. It's a moss, where I identified different polysaccharides with IF. So it turns out I've been working on this technique for about 10 years now, which is shocking to me to think about. So let's get started so we can talk about the workflow for today. So immunohistochemistry. It's a technique that uses antibodies in their hypervariable region to identify different antigens of interest in a tissue. And the antibodies that you use, they are able to identify a wide variety of different epitopes depending on what type of antibody you're using, what species of animal the antibody was made in, etc. But antibodies allow you to identify your target of interest, and they allow you to collect data about your protein expression, its distribution, and its localization. The workflow that I'm going to be going through today will start off with the extraction of your tissue from your animal model. And I'm going to be using brain sections on most of my slides just because that is the most familiar tissue for me right now at this point. But the techniques are applicable to all other tissues as well. But so the workflow starts with extraction of the brain and perfusion of the brain to have good quality of tissue. then making sections of the brain, mounting it, and then performing IHC. We'll go through an IHC workflow to generate a permanent product that identifies the location of where your antibody is binding to your antigen. And so I'm first going to start off with talking about perfusions to prepare your tissue, and then tissue collection and sectioning techniques. Perfusions are incredibly important. This is the beginning of your IHC process. If you don't have a successful perfusion or an adequate perfusion, this is going to impact the quality of your tissue right from the start. You need to, in your perfusion, have good clearance of blood from the body, and you need to have good tissue fixation. And so if you're using a mouse model or a rat model or a rabbit model or different size animals, this will require different volumes of fluid to, one, clear the blood from the body and as well fix the tissue. If you're using a mouse model, you typically won't be ligating or clamping any of the aortas, the ascending or descending. But if you're using a larger animal model, you might want to clamp the ascending or descending aorta to limit the amount of fluid you need to push through the body, whether that be the clearance media or the fixation reagent. The fixation reagent typically used is paraformaldehyde or formaldehyde at a concentration of 4%. And the way the perfusion normally goes is you'll have a needle that's connected to tubing on a peristaltic pump that is submerged in your clearance solution, which is PBS a lot of the time. You will insert that into the left ventricle, turn the pump on, clip the right atrium, and then the blood will flow out of the body to be cleared. Once all of the blood is removed from the body, you can switch to your fixative, paraformaldehyde, and fix your tissue. If you're teaching someone for the first time how to do perfusions and you have new technicians or students in the lab, you definitely want to over-exaggerate how much solution they need to use because their technique are for needle insertion as well as clipping the atrium for removal of the blood isn't going to be as good. So you want to increase the amount of fluid used to pretty much make sure you have full coverage of your tissue. So once you have perfusion complete, you need to remove your tissue of interest. So if you're removing the brain from the body, you need to get past the skin, you need to get through the skull. But once you have your tissue of interest, wherever that is out of the body, you can put that into a tube, which I will tell you what's in that tube in the next slide. But I want you to look at the picture where I have my gloved hand with the brain on it. You can see that it is not pink color like the brain above it, but it's like white colored. And the indication that there is blood left in the tissue is if there is any pink color left in the brain or any tissue you're using. So this is a good indicator that your fixation or your perfusion was not as successful and could indicate that you need to fix this tissue more post-perfusion and post-extraction from the body. So once you have your tissue out of your animal, you will then be putting it into a vial that's going to contain paraformaldehyde most of the time. And it's the same concentration paraformaldehyde, the same 4% that you used for the perfusion. And I usually leave the tissue in the paraformaldehyde overnight or up to a maximum amount of 24 hours. And this is for a good perfusion. And so I mentioned that if your perfusion was not as successful, let's say you still had some pink color in your tissue, you're going to want to extend the amount of time that the tissue is in the fixative agent because it's a high probability that the fixation was not as successful during the perfusion. And so you can extend that past the 24-hour mark up to the 72-hour mark, depending on how poor the perfusion process went. And so once you are done with that fixation step post hoc of your perfusion, you can then move on to preparing your tissue for sectioning. And there's two different methods that are generally used: preparing tissue for cryopreservation for cryosectioning, or preparation of your tissue with paraffin embedding, which doesn't require cryosectioning. It's just regular sectioning, not freezing. And so for cryopreservation, you'll see the two vials on the right. You're going to want to displace the water in your tissue with a compound. We use sucrose, and sucrose will go into your tissue, displace all the water in there. That will cryoprotect it, because water will freeze, so we don't want it in there. And when you first put your tissue in this 30% sucrose solution in the vial, it's going to float. And then 48 to 72 hours later is generally the time period I leave it in that solution. Your tissue will be sunk to the bottom of the vial, indicating that it has successfully displaced the water and now contains sucrose. On the other hand, if you're going to do paraffin embedding, what you're going to want to do after the fixation step is you're going to want to make sure you have all the paraformaldehyde rinsed off of it. And then you're going to want to dehydrate your tissue with a graded alcohol series from 70, 90 to 100% ethanol, and then into xylene. And once you've run through this series of solutions, you're going to place your tissue in a mold and then pour molten paraffin on it, and you let that cool, and then your tissue will be ready for sectioning whenever you would like. The cryoprotected method is one that I generally use all the time. And so I'm going to go into a little bit more detail on this method for this seminar. And so for cryoprotection, you're going to be using a freezing technique, and you're going to be using a frozen block of tissue for your sectioning. And so there's really one main method that people use for making these frozen blocks of tissue and OCT compound. I have my own method, kind of like a dirty get around method if you don't have molds, I'll tell you about on the next slide. But for the method that's generally used, the setup goes like this. So you have liquid nitrogen, or you have dry ice on the bottom of your beaker. And then on the next beaker that's floating on top of or sitting on top of the dry ice or liquid nitrogen, you're going to have an alcohol called methylbutane or isopentane on the inside of it. The reason you want to use this alcohol is because it's not going to freeze at really cold temperatures. It's going to stay liquid, which is what you want. And then on top of that, you're going to have floating a mold. It's usually a plastic mold. You can see it here as well. And you're going to have your tissue positioned in it, and you're going to fill this mold with OCT compound. And as soon as this sits on the freezing cold isopentane, it's going to freeze extremely quickly and make a mold of your tissue in this plastic container. Now, if you don't have the molds, I have a way to, or if you don't have liquid nitrogen, etc., I have a way to get around this. So the way to get around this is you can directly take your tissue and kind of move it into the cryostat after one key step. So I mentioned isopentane or methylbutane alcohol on the previous slide, and that's what was sitting on top of the dry ice or liquid nitrogen. So instead of using liquid nitrogen or dry ice to get it cold, what I do is actually I take like a 50 ml bullet tube and I put like 25 mils of isopentane in it. I'll stick it in the -80, let it drop to the -80 temperature, and then I will remove it from the -80, drop my brain into the alcohol for 30 seconds up to a minute maximum, remove it from the alcohol, put it into the -20 cryostat. Now at that point, the brain has been flash frozen because it was dropped into that -80 degrees Celsius isopentane, and now it's in the cryostat. Once it's in the cryostat, these like little metal pieces down here are the chucks that you can mount your tissue on in a cryostat. You take the metal chuck, you put a little OCT on it, it will freeze. You will stick your tissue of interest in whatever orientation you want down onto the chuck. And then once that's stable, you will then take a piece of tinfoil. Now this piece of tinfoil can be wrapped around your finger or a bullet tube, and it can be taped shut so it doesn't open, right? So you have a circular continuous shape. You'll stick that down on your brain or your tissue of interest, and then you'll simply fill up the foil so that the OCT is covering your tissue of interest, and let that freeze. And it'll freeze very quickly because your brain has already been, or just tissue has already been flash frozen, and you're in a -20 degree environment, and you will end up with this nice block of OCT that's ready to go already on a chuck for sectioning. So once you have your block for sectioning, there are a couple different things to consider. There's the thickness of the sections that you want to make, and there's different methods for collection of the tissue. So there are different thicknesses that you want to consider. There's thinner sections like 5 to 20 microns, and then there's thicker sections that are 20 to 40 microns. Thinner sections will have better penetration for IHC, but they are more brittle and they tear more easily. So if you're using thinner sections, I suggest using a roll plate or paintbrush method to directly mount your tissue onto slides. And these two methods basically produce a flat section like you see in this paintbrush method, or it would appear underneath this glass roll plate in the roll plate method. And you basically just take your slide and stick it down on that flat section. And you just keep doing that until you have as many sections as you want on your slide. And the reason you want to do this is you don't want to try to mount this thin tissue later because it will rip and it'll be more disastrous than you want to deal with. But if you have thicker sections, you can try the well plate method where you literally just can pick up with a forceps the rolls that will accumulate from sectioning and put them in a well that contains PBS. And you can keep this for a couple weeks at 4 degrees, or you can switch it to like cryopreservation media and put it in the -20 for later use. But two different methods to consider when collecting your tissue, depending on thickness or what you want to use them for. And so now you have your sections, you can finally start IHC. And so I'm going to go through antigen retrieval, blocking endogenous binding and activity. There's two types of blocking, primary and secondary antibodies. And so before you start your IHC, there are choices to make. So you may have free floating sections that you collected, or you might have direct mounted sections. You can do free floating IHC where your sections are in a well and you're going to add solution to that well and stain the tissue floating around in that well. Or you can do it mounted. And if you see in this bottom image here, there is a hydrophobic line drawn around these sections. And that hydrophobic line will prevent any solution from like leaving that area. And so the well plate method, it's better in the fact that you get full coverage of your sections on all sides of your tissue by your solution, but you need to use more antibody. You need 400 to 500 microliters per well for it to be sustainable. But if you use the direct mounted or already mounted sections, you use less antibody, but then your penetration for your tissue is limited to one side. So things to consider. But moving on to antigen retrieval. So antigen retrieval is allowing you to reverse some of the fixation that occurred during the perfusion and post-hoc fixation step. And this is because formaldehyde will cross-link different proteins in your tissue, and this might block the epitope that your antibody is trying to bind in your tissue. And so there are a couple different methods for antigen retrieval. It could depend on heat with citrate, TBS, or EDTA buffers, or it could be through enzyme digestion with like trypsin or proteinase K. And you really want to consider if this step is important or necessary. Some antigens and epitopes can be negatively impacted by antigen retrieval, so I highly suggest you do research on previous publications from other labs that have had success in using or not using this technique with your epitope of interest. And so moving on to the first blocking step. So the first blocking is blocking for non-specific binding. So in your tissue, the goal is to use your antibodies to bind to a specific epitope and only that epitope in your tissue. Well, it turns out that there might be other sites within your tissue that have similar structure or potentially charge that is similar to what your antibody is binding to. And so there could be some distractions for where your antibody binds. To limit the amount of distractions your antibody has from binding to non-specific sites, you will use blocking agents. And some of the agents are normal serum, which can be from the species of your secondary antibody, depending on what animal antibodies you are using, which I'll get into in a little bit. Or you could use bovine serum, albumin, or BSA. I've actually had great success just using BSA for at least the antibodies that I've been using. I just use a 1% concentration of BSA for my experiments, and I've had pretty good success for that. But this is definitely a step that can be troubleshooted if you're having issues with like increased background staining, for example. The second type of blocking is blocking for non-specific activity. So IHC depends on, a lot of the time, the activity of a specific enzyme, such as a peroxidase, to generate the signal of where your antibody is binding to the antigen of interest. And since tissue has endogenous enzymes in it that may still be functional even through your processing through cryopreservation or paraffin embedding, you want to get rid of that endogenous activity so that when you are giving your tissue substrate, the signal is only generated at where your antibody is bound and not some random location within the tissue. So for example, blocking endogenous peroxidase activity, you want to wear that enzyme out. So the way to do that is to give it its substrate. And you can give hydrogen peroxide. So you apply a 1% hydrogen peroxide solution to your tissue, and the peroxidase will pick that up, use it, make product, and wear itself out. And all of your endogenous enzyme activity in the tissue will then be spent. And that would be the goal before proceeding to applying any primary antibodies or anything like that. So moving on to antibodies, there are a couple of things we want to talk about. So first, we want to talk about the two types of IHC. We have direct IHC and we have indirect IHC. Direct is when you have an antibody, a single antibody that will have the signal molecule or, for example, the peroxidase attached to it so that it itself can generate signal without the need of use of any other enzyme or additive antibody. Where in the indirect method, you're going to be using two antibodies. You have a primary antibody that's binding to your antigen of interest, and then your secondary antibody, which is binding to your primary antibody. And your secondary antibody may have the enzyme of interest or could have the ability to attach an enzyme of interest to it to generate the signal that you would like. Next, there's also polyclonal and monoclonal antibodies. And so what are the difference between the two? So polyclonal antibodies, when you buy an aliquot of that, that's going to be a mixed population of antibodies. All of the antibodies are going to bind to the same antigen, but they're going to be binding at different epitope sites. This is beneficial in the fact that it would allow for more signal amplification of your signal on your protein of interest, because there's multiple different binding sites, you can get more antibodies in there. But if there's more epitopes, that leads for a higher possibility of more background staining, because a higher chance that your antibodies could be distracted by other epitopes that are similar within your tissue. Where you have a monoclonal antibodies for the other type, this is only going to be a single antibody species. So you're only going to have one type of antibody, and that antibody is going to bind to a single epitope on your antigen or protein of interest. And this is beneficial because less epitopes to worry about means there's possibly less distraction for your antibody binding non-specifically in your tissue. So monoclonals, a lot of the time you have lower background staining. But how are these two types of antibodies generated? They're generated in different methods. So if you're going to generate a monoclonal antibody, this is the more expensive generation. You're going to inject an antigen of interest that you want to make antibodies against into a model animal. From that model animal, after they mount an immune response to that antigen, they're going to extract plasma cells from it. They're going to fuse those plasma cells with tumor cells to make them into hybridoma cells, which are basically, they can survive and keep producing and replicating, and they're not going to die out. And you can then extract from these hybridoma cells specific monoclonal antibodies. Whereas if you are doing a polyclonal antibody production, you're going to have your antigen of interest injected into an animal. That animal is going to mount an immune response, and then they're going to receive a second injection of that same antigen. They're going to mount an immune response, and then you're going to collect their blood serum, and the result is a polyclonal antibody. And so that's for generating antibodies, primary antibodies against your antigen of interest. How are secondary antibodies generated? So making a secondary antibody, this is going to be against an animal species, not an antigen of interest. So what you do is you first will have to inject an antigen of interest into, for example, a mouse model. that mouse model will produce mouse antibodies against the antigen of interest. And then you will actually take those mouse antibodies and inject them into another animal species like chicken, goat, or rabbit. And the antibodies that you collect from these different animals will be called anti-mouse antibodies. So your secondary antibodies are always going to be anti-mouse, anti-goat, anti-rabbit, for example, because they're against the antibodies from another animal where the specific antigen generated antibodies are from. And so speaking of all those different species that you can have animals making antibodies from, you want to ideally choose a good combination of animal species for your antibodies. So ideally you want to use antibodies, and I've had the best luck with this, the least amount of problems. If you use antibody species from different animals than your tissue. So for example, a lot of the time I'm using mouse tissue, so I'm going to want to choose rabbit antibodies or sheep antibodies or chicken antibodies. I'm not going to really want to use mouse antibodies because you might run into more problems with that, because you'll have potential higher possibility of cross-reactivity. And so that's all we're going to talk about with the antibodies. So now let's talk about the generation of signal, the actual stain that appears in the tissue, counterstaining after you are done with generation of your specific signal, and then mounting your tissue. So for generation of signal, what you're hoping for is a permanent product appearing specifically where your antibodies are bound and not in non-specific sites within your tissue. And as I said before, this reaction typically relies on enzymes in the tissue, but not the endogenous enzymes. It's going to rely on enzymes that might be attached to your secondary antibody, or you're going to want to attach them to your secondary antibody in a successive step. The most common enzymes that are used for generation of signal are going to be horseradish peroxidase, which is going to convert DAB into a brown permanent product, or you're going to have alkaline phosphatase, which is going to convert AUC into a red product. And both of these are insoluble and they are photostable. So if you want to, for example, look at slides that were in your lab that you're in right now from 20 years ago, they're probably going to still look just as good as they did 20 years ago, of course, if they were cover slipped well and all else considered. But very permanent and stable product. You can image it over and over again and it's not going anywhere. And so for chromogenic detection, for generating your signal, there's two main methods. There are biotin-based methods and there's non-biotin-based methods. So I'm going to go over the biotin-based methods first. And so biotin-based methods, they are like, depending on the binding between avidin and biotin, or streptavidin and biotin. Avidin and streptavidin, they're only different in the fact that streptavidin lacks a glycoprotein, and the fact that it lacks that glycoprotein allows it to be less non-specific binding. It has less electrostatic interactions than the regular avidin would have. So if you see those two names pop up when you're looking for antibody reaction kits, that's what the difference is between those two. But basically, you're using that avidin biotin complex to attach your enzyme of interest to your secondary antibody, for example. So we have our antigen, we have our primary antibody, and we have our biotinylated secondary antibody. And our biotinylated secondary antibody itself does not have the enzyme we want on it, for example. It might not have the enzyme. So we want to attach horseradish peroxidase to it so we could do a DAB reaction. And that's where streptavidin comes in. All these pink, like light purple highlights on there, those are the horseradish peroxidase enzyme. And you are using your biotin on your secondary antibody and biotin bound to streptavidin to attach that horseradish peroxidase to your secondary antibody. And that's how you're going to get the specific reaction only occurring at your antibody location. And so the downside to the biotin method is that there is endogenous biotin in your tissue. And so if there's a lot of endogenous biotin in your tissue, you could have some distraction on where your streptavidin is binding, meaning your enzyme would be in different places that you don't want it to be. And if you do antigen retrieval, sometimes this will actually increase the prevalence of biotin epitopes in your tissue. So you want to consider that as well if you're using this method. And so if you're having trouble with high background staining using this biotin method, I suggest trying the method here on the next slide, which is non-biotin based. So the non-biotin based method, this one relies on the original method was developed with a polymer backbone. And you can see over here, we have antigen, primary antibody, secondary antibody in green. And then this purple is this polymer backbone, like made of dextrin, for example. And the red are the enzymes attached to that. And so basically you're using this dextrin backbone as a bridge for your secondary antibodies and your enzymes to bring the enzymes to your site of interest. The issue with this dextrin backbone system is that it's really sterically hinders everything because it's so big. So you can't really get as many antibodies in the same location, meaning you have less signal amplification. And so they actually generated a new method which contains polymerized enzymes on the secondary antibodies. So we have our primary, we have our secondary, and then instead of that giant dextrin backbone, it just has a lot of enzymes that are polymerized together now. And this helps get around that issue of less signal amplification. So definitely try this method out if you have had poor success with the biotin method. And so once you've generated your signal, you want to move ahead and counterstain your tissue potentially. Counterstaining allows you to identify structure of tissue to help localize a nucleus of interest in the brain or a different region in a different tissue, because, for example, if you're going to stain for nucleic acids, cells are packed differently in different locations, meaning they're close together in some places, they're far away in some places. You can see here, this is the example of the hippocampus in the brain. You have this nice band of nucleic acids of the nuclei of where the neurons are really packed close together. And that helps you identify, like, that is the hippocampus. Okay, I want to localize my target, my images that I'm going to take to that region. And so there's a bunch of different counterstains you can use. Shown in this image here are hematoxylin and eosin stain. Hematoxylin identifies nuclei, and this is blue to violet color. And eosin identifies the cytoplasm, which is pink to red. Those are the most common ones. You can also use fast red, which appears red. It's also for nucleic acids, or methyl green, which is also for nucleic acids, but it shows up as green instead of red. And you might want different colors depending on what type of chromogenic stain you're using for better contrast for imaging and localization of your protein. And so depending on what method you chose to stain with, if you chose the method where you use the hydrophobic pen on a slide and your sections were already on a slide while you were staining, you don't need to worry about mounting. But if you were doing free-floating sections, you're now done with staining, and now you need to mount them in order to cover slip them and preserve them for imaging after your experiment is done. And so I'm going to show you a video of a method that I have the most success with preventing damage from occurring on your tissue during the mounting process. And so what I do is I put a slide down in a well that has PBS in it. And I actually pick up those brain sections and with a paintbrush, and I push them down on this positively charged slide. Just stick them down, and then I pull the whole slide out of the PBS, and they all stick together pretty well on there. And I mean, of course, this is going to take some, trial and error, and it's going to take some practice, but I really like this method because having them float down onto the slide itself is actually preventing you from touching it as much. And you aren't going to have as many rips in your tissue or distortions or folds in your tissue as you might get if you're just mounting your tissue on a slide on a bench and using like a couple paintbrushes to flatten out your section. So maybe try this method out if you haven't before. I really like it, and I use it for all my experiments, typically if I'm doing free-floating sections. And so once you are done mounting, you can move on to the alcohol dehydration and the xylene clearing steps. And so when you are doing a chromogenic stain, you might have some extra stain in your background on your tissue that you don't want to be present, and the alcohols actually help remove that. And so you're going to be pushing your tissue through 80, 90, and 100% alcohols. I usually do 2 minutes in each of these baths, but if I want to remove more of the background staining for better contrast of my stain on the specific sites where my antibody is bound, I'll actually leave it in the 80% a little longer, and you'll get a little bit more of that stain coming off. So if you look at this little video here, you'll see as I pull it out, you see all the purple that is left behind in the well. And if I wanted to remove more of that purple, I would just leave it in the 80% a little bit longer. But you kind of have to play with this depending on what type of stain you're using or how susceptible your staining procedure is to the alcohol steps. You kind of got to play around with it, but I suggest staying with 2 minutes to start, and you can do less or more from there. But once you're done with the alcohol steps for dehydrating your tissue, you're going to want to put your tissue in xylene. And then you do that for about 5 minutes, and then you can cover slip directly out of xylene. And you're going to want to use a non-aqueous mounting media, of course, because your tissue has been dehydrated and placed in xylene now. It is no longer aqueous miscible at all, so non-aqueous mounting media. And so now on to some controls that you might want to consider using for your experiments. So we first have antigen controls. And for antigen controls, we have positive controls, we have negative controls, and we have endogenous tissue background controls. And so for positive controls, this is when you are using a type of tissue that is known historically based on evidence from other labs, publishing papers, etc., to have your protein of interest in it for sure. So that regardless of the result of your experimental tissue, if your positive control tissue works, you know that your reagents are working properly, and it's just an issue with your experimental tissue that you need to figure out. For an example here, I have an image of the hippocampus. And the hippocampus, if you're looking for a protein called CFOS as an immediate early gene that's expressed when neurons get activated, the hippocampus is pretty active a lot of the time in the brain. So I use this region as a control region for a positive control for CFOS protein. And so even if my experimental tissue doesn't show CFOS, but the hippocampus does show CFOS, I know that my reaction is working, and it's just an issue with my experimental tissue. The negative control is kind of the polar opposite. You want to use tissue that is known to not express your protein of interest to check for any non-specific signal. And lastly, we have the endogenous tissue background control. So this, you legitimately just want to take your tissue that is unstained, cover slip it, put it under the microscope, check for any pigmentation like you would see here if you're doing a chromogenic stain, or if you're doing IF, you want to check for any endogenous fluorescence that is present. You want to be familiar with what your tissue looks like before you stain it, because you might be distracted and think that's a positive signal. So I highly suggest you do this for, especially for a new tissue that you have not worked with before. And so the second controls are reagent controls, and these would be no primary antibody. These would be isotype controls, or this would be absorption controls. And so no primary antibody, I highly suggest running with your experiments all the time. It's when your tissue is incubated with everything in the protocol except for the primary antibody. And what does this do? It tells you that none of your other reagents are going to create any non-specific staining in your tissue, that the staining is specific for binding to where your primary antibody is bound. The next one, which you might not be familiar with, is this isotype control. It's when you use the antibody of a same isotype. So antibodies are, there's IgG, IgM, IgE, IgA. They have different clonalities, conjugates, and host species, same isotype, to see if there's just any non-specific binding of that antibody within your tissue on a regular basis. And so I suggest using this as a troubleshooting control. You don't need to do this right off the bat, but if you're having issues with staining, maybe try this control. And lastly is the absorption control. So this, you want to pre-incubate your primary antibody with the antigen of interest in a tube. And then once the incubation is done, you want to put it onto your tissue of interest and run your IHC reaction. And you should not have any staining because your primary antibody should already be bound up with its antigen of interest. If you see any staining, that's telling you that your antibody is a little non-specific, and any staining you see are regions that it bound that you don't want it to be bound to. Okay, so some actual troubleshooting slides. So the problem on this slide would be low signal detected. So you see it on this top image. Both of these images are supposed to be the same neuron set. The top image, you can barely see any staining in this region, but the bottom, lots of staining. You can see dendrites, you can see somas of the neurons there. And so this is a troubleshooting step that I had to figure out myself because this is tissue that I use for my experiments. And so ways to improve this is you can use a higher concentration of primary antibody. Make sure you do an antibody dilution series to see the most proper concentration of your antibody you're using. You can incubate your sample for a longer period of time with your primary antibody, and definitely make sure you're running a positive control to determine if the signal is just bad on your tissue or if the reaction in general is not working. Next, we have poor definition of structure in tissue. So on the left here, we have neurons that are lacking really good definition of their dendrites and axon structures coming off of the soma. On the right here, we can see lots of definition of dendrites and the axonal structures. And so ways to improve this that I've found were useful, because I had to troubleshoot this as well for my stuff. So you can extend the blocking step, make it longer, or you can use different serum. Potentially, if you're using BSA, switch to an actual serum. This might be a resultant of the tissue not being fixed well during perfusion. So unfortunately, this would be back at the very beginning, leaving you with poor tissue quality. And you can also reduce the time spent in the alcohol dehydration steps before you are cover slipping. I've seen that by limiting the amount of time in alcohol, this has actually improved this for me. So if you're doing any neuronal structures, you might want to consider this, or any fine structures in general. And lastly, we have a problem of high background staining. So on the top here, we have dark tissue. On the bottom here, we have lighter tissue, but we have still dark staining of the region of interest of where the neurons are. And so ways to improve this is to check your secondary antibody to see if it was binding non-specifically. So run that primary antibody control, remove it from the reaction, and just see what your secondary antibody is doing with the rest of your reagents. You can again increase the blocking of non-specific binding time or change the serum. Your primary antibody concentration might be too high, actually, so you would probably want to dial it back and dilute it a little further. Or it could indicate your washing steps aren't sufficient. You're probably cutting some time out of your washing steps. Don't do it. Add another wash step if you're hesitant about this, or extend the washing time. The more solution changes you have, the better off you're going to be. And also, if your tissue gets dried out before your experiments at all, this is going to be an issue, and it's not going to leave you with good tissue quality for your experiments. So keep them in wells, keep them in liquid, or keep them in a humidified chamber. And so that's all for the run through, and I'd be happy to take any questions after Afrida goes through a few more slides.
All right, well, great. Thank you so much, Elizabeth, for another wonderful workshop. As she mentioned, I do want to just share a couple product slides that might be relevant for some of the audience members here if you're trying to optimize your IHC workflow. So Proteintech does offer a wide selection of products that can help you with your IHC workflow, including our primary antibodies for IHC. So we have nearly 9,000 antibodies that have been demonstrated to be successful in IHC, and we offer both unconjugated antibodies and biotin conjugated antibodies. We also offer IHC kits, which I'll talk a little bit about in the next slide. And we also have supporting products for IHC. So these will be your antigen retrieval buffers and your hydrophobic pens. So as I mentioned, we offer IHC kits, and these come in 3 different flavors. So we have the IHC Easy Ready to Use kits. And so these kits are great if you are beginning IHC or you just had a committee meeting and your committee asks you to look at a very particular protein and you just don't want to deal with the troubleshooting. So these kits are great because we've done the troubleshooting for you already. So the kits are target specific and they're ready to use. They contain the primary antibody already, along with all the reagents that you would need in order to complete your IHC. We also provide an easy to follow protocol, and our kits do cover a very broad range of targets. We also have our IHC prep and detect kits, and these are great if you're trying to develop an IHC workflow where you already have a primary antibody that you like, or you have one that you really want to use. So these kits contain all the basic reagents that you need to do your IHC without the primary antibody being included. So these kits are tested in-house with multiple primaries so that we have validated that they work very well. We also have our IHC detect kits, and our IHC detect kits are great if you already have a protocol for your tissue preparation and you really just need something for high sensitivity to look at your chromogenic signal detection. So this is great for looking at your signal. And again, we test these kits in-house using multiple primaries as well. So I just wanted to show some quick data on our IHC Easy kits. So all our IHC Easy kits are optimized for the primary antibody dilution, and then this dilution is confirmed in various tissue samples. So this is an example of our beta-catenin, and you can see that we have optimized the dilution to 1 to 20,000, and then confirmed this in colon tissue in 3 different species. And finally, I just want to share that we do have supporting products for IHC, including antigen retrieval buffers, whether you're using heat-induced or proteolytic-induced epitope retrieval. And we also provide hydrophobic IHC pens. So this is great if you are trying to prevent liquid from running off the edges of your slides. So as a attendee of today's webinar, you are eligible to receive a free sample and also a promo code for any IHC kit. So our free sample covers any unconjugated Proteintech antibody, and our promo code IHCWeb25 is 25% off of any IHC kit. We will be providing a follow-up email that will have all this information for you. So with that, I will go ahead and begin the Q&A session for Elizabeth. So it seems like we have a lot of great questions. So someone asks, when you perfuse with PBS, do you prefer with calcium, magnesium, or without?
So I typically use it without calcium, magnesium, and it's perfectly fine.
Okay. So to produce a secondary antibody, so I think they mean to use. So to use a secondary antibody, can you use rat as a different type of animal from mice, or does it have to be more different of a species like sheep or rabbit or rooster?
So using mouse antibodies against rat or rat antibodies against mice, it definitely can work. It just, they are closer in species than the other animals. So I have had some issues using rat against mouse or mouse against rat. So if possible, use a different species, but if you can't, that's fine. You just need to make sure with controls that your reaction is running appropriately.
Great. Someone's asking, can we do antigen retrieval for frozen fixed tissue?
So you can do antigen retrieval for your frozen tissue. It is highly dependent on the target you're looking for. So for an example, since I mentioned CFOS previously on one of the slides, that's a pretty that's a protein that kind of goes away really easily if it's exposed to heat for too long. If it's left for room temperature for too long, it's not going to be present for the epitope that you want. So I would definitely do background reading for your antigen specifically to make sure that your antigen is compatible with antigen retrieval.
Right. So someone's asking, this is a question for Proteintech, if we customize IHC ready-to-use kits for individual needs. Currently, we don't, but this might be a case-by-case basis. So if you want to email me, I will make sure and get your information to the right people who can discuss this with you. So someone's asking, how much antibody does the mounted method use?
In terms of volume, I'm assuming not dilution, right?
I'm assuming it means volume, yes.
Okay. So yeah, I mentioned that in the well plate, you need like 400 to 500 microliters. You could probably, so it depends on how many sections, of course, you're staining on your slide, but if you're doing a single section, you drew a hydrophobic line around that single section, you could probably get away with like 50 microliters and you'd be fine.
So this is a good question. So they're asking, if you need to store tissue longer, for a longer period of time before paraffin embedding, what's the preferred method of storage?
So before paraffin embedding, you're definitely going to want to get your tissue cold and get it into the -80 for long-term storage. I would probably suggest flash freezing your tissue and not just putting it in the -80 because again, any slow freezing technique is going to create like cracking artifacts in your tissue. So flash freeze it and then get it into the -80.
So there's someone who is saying that they're using a mouse antibody to see mouse tissue sections because it was mentioned that they can use it for mouse tissue. So do you think that they should still prefer another host, or should they continue using that mouse antibody?
So no, absolutely. Like, so if you have mouse tissue and you have a mouse antibody, it definitely can work. I just tend to avoid it off the bat if I can, if the antibodies are around, because you don't have to worry about as much cross reactivity. But of course, you can use the mouse antibody. Just again, run controls to make sure that you don't have non-specific binding anywhere in your tissue. So definitely make sure your positive control is working correctly.
Okay. So there is an IF question, which you are also very familiar with IF. So someone's asking, when using multiple antibodies to detect fluorescent proteins, 4 of them, on mouse tissue, there's a lot of background sometimes, and there is cross-reactivity amongst them. Do you have any suggestions?
So, okay, when you're doing IF with multiple antibodies, so you have, different antibodies against different antigens, and you want to get multiple different fluorescent signals working together, I suggest using secondary antibodies from the same animal for all of them if you can. So what I do is like I have I have like a chicken antibody and I have a goat antibody, but then my secondary antibody is both donkey for both of them. And that tends to eliminate any other types of non-specific binding you would get with secondary antibodies from different species. So try to use the same animal for your secondary.
Yeah, and kind of to add to that, the other thing that you could really try is using conjugated antibodies and kind of doing a direct IF instead of
Absolutely.
indirect. You could also, if you can't find a pre-conjugated antibody, you can also label your own antibody. So Proteintech actually recently launched our flexible antibody labeling kits. So if you don't have an antibody that is already conjugated, you can conjugate it yourself as well. So there is, someone's asking to clarify, I think this was the question about the amount of antibody on the slides. So they're saying to clarify, do you add 400 microliters of antibody per well, or is the 400 microliters in amount including PBS?
So the 400 microliters is your your solution that your antibody is diluted into, right? So this might contain Triton, PBS, BSA, and your antibody that's been diluted in that solution down to like 1 to 10,000 or something, for example. So like a 1 to 10,000 primary antibody solution containing all those other components is in the 400 microliters.
Great, thanks. So someone's asking, is there a way to perfuse sections of an organ if you're not able to perfuse the entire organ?
So I guess you have to take the tissue out of the animal before, or like, let's say the animal dies and you have to quickly remove tissues from an animal. Sometimes that happens. The way that we kind of deal with this is we will take that tissue, try to preserve its integrity while taking it out as best as possible, and then submerging it in 4% paraformaldehyde for up to a week. It's not going to be the best perfusion, but you will be able to fix the majority of it, especially if it's a thinner tissue. If it's a thick tissue, it's more difficult with that technique.
Yeah, I was going to say it probably is dependent on the organ, because it might be difficult if it's brain, then maybe not, because you really need to flush out all that blood.
Yeah, I actually have done that. It's just it's not the best tissue quality. So if you're doing IHC with it, it's not going to be the best quality. So, but it is possible.
All right, I think we have time for one more question. So do you have any advice or a preferred method to analyze the colocalization data from double and triple IF?
So if you don't have access to like fancy microscopes or fancy software that comes with the microscopes for colocalization, you can, if you just want to go use ImageJ, you can try using ImageJ. They have a cell counter there, and they also have the ability to easily turn off colors and turn off colors if you're just starting with this type of technique. But it's obviously preferable if your lab has access to a higher power software that comes with microscopes a lot of the time. So it's really dependent if you have access to better software or not.
Great. Well, thank you. Thanks everyone for all the questions. So there are a lot of questions we didn't get to, and Elizabeth has graciously agreed to answer those offline. So I will send her a list of the questions we did not get to, and then we will email her response to you guys. But I want to just quickly thank Elizabeth for not hosting this once, but twice for us, and both times were amazing workshops. She put a lot of work into this, and we're really appreciative of that. And we're also really appreciative of everyone who joined in today. You guys had really amazing questions, and we really appreciate that. So definitely watch out for that follow-up email that will have our free sample link and also our promo code for 25% off of any IHC kit. So thank you again, everyone, for joining, and have a great rest of your day.
Have a great day.